Hello, my name is Nidia Trovão, and I'm a researcher at the Fogarty International Center at NIH. I am a molecular epidemiologist of infectious diseases, working with genetic data to investigate the different aspects of viral evolution, spread, and adaptation. In this section, we are going to talk about how to reconstruct the spatiotemporal diffusion of a pathogen in a continuous space. More specifically, this tutorial provides a step-by-step explanation on reconstructing the spatial dynamics of yellow fever virus epidemic in Brazil based on a set of viral genome sequences isolated at different points in time. The yellow fever virus is responsible for 29,000 to 60,000 deaths annually in South America and Africa and is the most severe mosquito-borne infection in the tropics. The viral transmission occurs via the salvific cycle, in which humans and non-human primates are infected by mosquito vectors. Transmission can also occur via urban cycle, in which humans are infected by Aedes species mosquitoes that feed mostly on humans. Recently, Brazil has experienced its largest recorded yellow fever outbreak in decades. Our aims are to characterize the dispersal history and dynamics of the virus during this recent outbreak, estimate the ancestral dates and locations of the virus in continuous space, and visualize the spatiotemporal dynamics. We are going to analyze 65 whole genome sequences of the yellow fever virus with 10,236 nucleotides in length, sampled from humans and non-human primates in Brazil, with collection dates from throughout 2017. Now let's go over how to set up and run these analyses. To load the alignment into Beauty, simply select the Import Data option from the File menu and browse to the yfvsequences.fasta file to load it. The sequence data will be listed under partitions as shown in the figure. To set these dates, switch to the Tips tab at the top of the window. In this case, the yellow fever virus sequences have been sampled at various times during 2017. So let's select the box labeled Use Tip Dates. The actual sampling time in years is encoded in the name of each taxon. So a convenient way to specify the dates of the sequences in Beauty is to use the Parse Dates button in the top left. Select Define by a prefix and its order, then select Last from the drop-down menu for the order, and set the prefix to the pipe symbol. For the yellow fever virus sequences, date values have to be parsed as calendar dates by specifying the format day-day, month-month, and year-year-year-year. Press OK, and the dates will appear in the appropriate column on the main window. So you can now check the dates and edit them manually if needed. The height column lists the ages of the tips relative to time zero, in our case, 2007.304. The uncertainty column allows specifying the precision of the sampling time. This is not useful in our case because all sampling dates are known to the exact day, but BEAST allows integrating over the uncertainty for less precise sampling dates, like when only the sampling month or year is known. The next thing to do is to specify the traits. A trait can be any characteristic that is inherent to the specific taxon, for example, geographical location or host species. This step will assign a latitude and longitude as a bivariate geographical location to each taxa based on the trait specification for each sequence. Next, click on the Traits tab at the top of the main window. Click on the Import Traits button, then browse and open the coordinates file. After clicking OK, select both the latitude and the longitude traits in the left window and click on the button Create Partition from Traits. In the window that pops up, enter a name for these partitions, for instance, location. This new location partition with two sites and with continuous data type will be shown under the Partitions tab. Next, click on the Sites tab in the top of the main window. This will reveal the evolutionary model settings for BEAST. Exactly which options appear depend on whether the data are nucleotides, amino acids, or traits. This tutorial assumes that you are familiar with the available evolutionary models. However, there are a couple of points to note about selecting a model in Beauty that can be reviewed in the Estimate Dates and Rates tutorial. For the nucleotide substitution model in these analyses, keep the default HKUI substitution model, keep the base frequencies to be estimated, specify the site heterogeneity model to gamma, and keep the default partition into codon positions off, since we are dealing with the full genome dataset with stop codons and regions that might not be in the correct open reading frame. Next, click on Location in the Substitution Model window, select the Cauchy Relaxed Random Walk model, and select Bivariate Trait representing latitude and longitude. This option generates diffusion statistics that are specific for bivariate spatial traits. Also, select the Add Random Jitter to Tips, which adds noise drawn uniformly at random from a particular jitter window size to duplicated location traits. Set the jitter window size to 0.01. The noise avoids a poor performance of the Brownian diffusion models when not all sequences are associated with unique locations. The molecular clock model options in the Clocks tab allows choosing between a strict and a relaxed clock. We will perform our run using the log normal uncorrelated relaxed molecular clock. Now move on to the Trees tab. This panel contains settings about the tree. First, the starting tree is specified as randomly generated. The other main setting here is to specify the tree prior, which describes how the population size is expected to change over time according to a coalescent model. The default tree prior is set to a constant size coalescent prior. Here we will select a flexible Bayesian skygrid coalescent model as the demographic tree prior with 36 grid points as the number of parameters and the time at least transition point set to 0.69. By doing so, the grid points actually approximate the number of epidemiological weeks spanned by the duration of the phylogeny. In the States tab, check that the location partition is set to reconstruct states at all ancestors, which is usually the default. Now let's review the prior settings under the Priors tab. We can keep the default priors in this case since BEAST provides priors that should accommodate most inferences. However, one should always search the literature for more informative priors when available. The Operators tab in Beauty has a table that lists the parameters, their operators, and the tuning settings for these operators. As mentioned in previous tutorials, the operators specify how the parameters change as the MCMC runs. In this tutorial, we will use the classic operator mix. The MCMC tab in Beauty provides settings to control the MCMC chain. For details on how to set the MCMC tab, refer to the tutorial on evolutionary rates and temporal estimates. For this dataset, let's set the chain length to 500 million and both the parameter logging to file and state echo to screen every 50,000 states. The next option allows the user to set the file stem name. Set these to YFV_Cauchy_RRW for relaxed random walk Cauchy diffusion model. The next two options set the names of the log and trees files. These will be set based on the file stem name. We can also log the operator analysis to a file. So, at this point we are ready to generate a BEAST XML file. To do this, either select the Generate BEAST File option from the File menu, or click the similarly labeled button at the bottom of the window and save the XML file. Once the Beast XML file has been created, the analysis itself can be performed using Beast. Press the Choose File button, select the XML file you just created, and click Run. If Beast was called through the command line, then the name of the XML file is added as an argument after the Beast command. When clicking Run, the analysis will be performed with detailed information about the run progress being written to the screen. When finished, the log and trees files will be saved in the same directory as your XML file.